Myself, Dr. Manvendra Singh, Department of Biotechnology, Faculty of Engineering and Technology, KMCL Language University. So, I am from biotechnology, so my topic is cell biology. First of all, we want to know what is cell. Cell is the basic unit of life, also defined as structural and functional unit of life. Cell is structural and functional unit of life. First of cell, 3.5 centuries years ago, approximately 3.5 century years ago, scientists invented their lenses, their microscope. So they invented a microscope and saw a dead cell. Why dead cell? Because at that time cork used for closing the bottle. So that piece used in microscopic study. So he saw cell. So he saw hexagonal structure and termed coined cell. How termed coined cell? At that time, Buddhist monk live in hexagonal shape structure and prayer for meditation state. So this structure known as at that time cellula. So from cellula, Robert Hooke termed that coined cell in 1665. After many years, more scientists search in this field, new terminology, new theory, and 1838, a scientist M. Sheldon, he was the botanist from Germany. He gave a statement: plant cells are made up of cells. When he saw microscopic study of cells, plant cells, he is the botanist. So we know the botanist interested in plants. So plant cells are made up of cells. He gave the statement 1838. When next scientist in 1839, after giving the statement, plant are made up of cells, T. Swasasang, so he was the zoologist from Britain. Zoologist also study the animals, and he gave three statements: plant and animal cells are made up of cells. And another statement: animal cells are covered by plasma membrane. And the third statement: cell wall present in plant cell. So first statement from M. Sheldon from botanist Germany: plant are made up of cells. And 1839, Swan, zoologist from Britain, he gave another statement: animal and plant are made up of cells, and animal cells are covered with plasma membrane, and cell wall present in plant cells. So complete theory 18 Sheldon and Swan. But another statement also included in this theory, in cell theory, the complete statement from 1838, 1839 and 1855. One scientist, Rudolf Virchow, he stated here their statement: omnis cellula e cellula. Omnis cellula e cellula. After we know plant are made up of cells, animal are made up of cells, animals covered with plasma membrane, and cell wall present in plant cell. These statements, after study these statements, so Rudolf Virchow gave a statement: omnis cellula e cellula. Omnis cellula e cellula means cells come from pre-existing cells. Cells come from pre-existing cells. This is the complete cell theory from 1838 to 1855. After we know about cell and cell theory. So we going to study different type of cell. So cell are divided in two parts: prokaryotic cells and eukaryotic cells. Prokaryotic cells means lack of nucleus, and eukaryotic cells means well developed nucleus and well developed structural organization. Cell organelles are found in eukaryotic cells. When we study prokaryotic cells, we study bacteria. General characteristics of prokaryotic cells: they have they lack nucleus, so they have circular form of DNA, circular form of genetic material found in prokaryotic cell, which float in cytoplasm of bacteria. So this is the circular DNA genetic material. Prokaryotic cells have contain one chromosome and extra chromosome, but eukaryotic cells have, like we are humans, so we have 46 chromosomes, but in prokaryotic cells single chromosome. So this is circular DNA. This is also known as nucleoid. Nucleoid means not well developed nucleus genetic material. So in prokaryotic cells, single circular DNA known as nucleoid, and extra chromosomal DNA known as plasmid. Plasmid, prokaryotic cell have one plasmid and more than one plasmid. So the plasmid is extra chromosomal DNA. So in this series, the structural organization of prokaryotic cells, layer covered by plasma membrane, outermost layer plasma membrane, which are found in all prokaryotic cells, and covering of plasma membrane covered by cell wall. Prokaryotic cells, cell wall of bacteria is made up of peptidoglycan. Peptidoglycan also made up of their single form peptidoglycan made up of NAG and NAM. NAG and N-acetyl glucosamine, N-acetyl muramic acid. So cell wall made up of peptidoglycan, and the outermost layer is glycocalyx. Glycocalyx not commonly found in all prokaryotic, all bacteria, but these are general characteristics. So we are count here as a part of eukaryotic. So glycocalyx also known as capsule, some places a slimy layer. So the glycocalyx. In this when in the series we discuss about prokaryotic cell organelles, when plasma membrane enfold inside and have different enzymes, vesicle, tubules. So these structure known as mesosome. And another place in cyanobacteria, plasma membrane enfold inside, and this structure have pigment, different type of pigment. So prokaryotic cells lack of chloroplast, so they not have different cell organelle. So pigments are here in cyanobacteria. So this structure known as phytochromo, analogous to chloroplast, and this is mesosome, analogous to mitochondria. And nucleate when generate developed RNA. So prokaryotic cells have 70S ribosome and eukaryotic cells have 80S ribosome. When 70S ribosome attached with the RNA strands and more than one ribosome attached with RNA strand, this structure is known as polysome. Polyribosome. So more than one ribosome, 70s ribosome. 70s ribosome also divided in two parts. '50s ribosome and 30s. 70s ribosome have two units, 50s and 30s. So, and prokaryotic cells also have a motility structure. This is flagella, pili, pilus. These structures are also found in prokaryotic cells. Flagella, pili, fimbri, and some structure stored as a nutrient in adverse condition, glycogen granules, phosphate granules. When phosphate granules are available in their environment, bacteria proliferate, producing in higher quantity. So these phosphate granules and glycogen granules combinedly known as inclusion body. So these are the structure of prokaryotic cells, and we will continue in next lecture.